So good evening to all of you. So how was the day? Good. Himanshu is always, energetic, right? Yeah, hope you did a RNA isolation. So I don't know how far you reached. Did you quantify? Okay. We will take up the results in a separate sitting, right? So you have to run the gel also, right? So that maybe perhaps we will be doing tomorrow. Okay. Yeah, students, we are assembled here just to attend another lecture by a practicing genomic scientist, right? A bioinformatician and a trained bioinformatician. So working for a company in Bangalore and a rich expertise in genome assembly. Okay, you can ask whatever clarifications, doubts you have on genome assembly. Whether it's how to assemble Illumina reads, how to assemble PacBio reads, how to assemble both Illumina and PacBio reads, what is called as hybrid assembly, right? And feel free to interact with the speaker. So I just request those who are sitting on the back side and come and sit in the first row. And let us complete the first row from here to here. Ramesh, welcome back. I think you had a high profile meeting this morning. So Ramesh and one more. Who is another person who accompanied you? Where is Darshan, by the way? Okay, they are still in the high level meeting. Okay, good. That's very nice. No? Students attending high level meeting is really appreciated.
You have a pointer, right? If you want me to remove this, I can do that.
This is pointer, this is forward, this is reverse. Forward, reverse.
And the middle one is the pointer?
Pointer, yeah.
Okay. Can I change it and see whether it is doing?
So you can take down the notes also. And Arpita's lecture is already given in the manual.
It's moving as it is, right? Yeah. कोई और बटन तो नहीं है ना?
नहीं वही दो है.
मैं इसको सेव कर दूँ. मैं डेस्कटॉप पे ले लूँ क्या?
क्या चीज़?
डेस्कटॉप पे सेव कर दूँ.
So all of you experienced quantic assembly, correct? It was demonstrated here, rather you got training also on quantic assembly. And Arpita's lecture would supplement that. And she will be talking about genome assembly. Okay. Students, I take pleasure in introducing Arpita Ghosh, She is a manager bioinformatics at Eurofins Genomics India Private Limited, a company who is involved in genome sequencing, transcriptome, or you name any omics service. They are ready to offer their service to you. And she is associated with this company for the last 3 or 3.5 years. Prior to taking this job as a manager bioinformatics, she was involved in another company, that is Acceleris Labs, that is in Ahmedabad, again as a bioinformatician. So that only speaks about her rich experience in the field of genomics, in particular the genome assembly. And she is a trained bioinformatician. I think she did masters in bioinformatics.
Yes.
And later on she took degree for business management as well. So the combination of bioinformatics and business management made her business manager or manager in bioinformatics at Eurofins. Okay. I invite Dr. Arpita Ghosh to deliver her talk on genome assembly, and I'm sure this lecture will in the sense enlighten you, rather update you over and above what you learned in the last 7 days. And she'll be talking in length about the strategies for assembling genomes. I think last year I requested her to give a special focus on microbial genomes, and she did a wonderful job of delivering a lecture here, well appreciated by many. And that only prompted us to invite her once again for addressing the student trainees here. And I thank Arpita for accepting our invitation, and I'm sure you are going to enjoy her lecture. Yeah, please.
Thank you. Yeah, I can stand here itself. Thank you. Is the mic on? Yeah. Thank you for a wonderful introduction. I am a trained bioinformatician, and I will talk about the basics, how we go for a genome assembly, or what we should consider when we are going for a genome assembly. So, the basics of plant pathology is that we have bacteria, viruses, fungus, everything infects plant. What is the common thing is the genome assembly. Genome sequencing of any of these organisms can get you into many details about these organisms and how you can understand the plant and the host pathogen interaction you can understand. So, these applications of genome assembly is very important. Genome assembly is just a technique how we do it, but how we interpret and get to the depth of science, that is what is important in understanding and to interpret and get to a result. The main applications of plant pathogens are, we can identify the pest which is infecting. For example, I have more than one organism infecting which is non-culturable bacteria. If it is non-culturable, I cannot identify with the traditional techniques, right. So, we also have DNA, we can isolate the DNA from that particular sample and identify which bacterial species, whether it is a non-culturable. If we do not know which bacteria it is, still we can come to a conclusion that this is getting infected with a non-culturable bacteria which is cannot be cultured. So, we can do a DNA sequencing and identify that, right. So, if it is more than one also, we can do metagenome and identify. So, that is what is the strength of NGS there, right. NGS can do all this. Sanger sequencing, which we have taken classes in different sessions, Sanger sequencing cannot do when you have more than one species, but NGS can do it and identify that which organism it is. If not which organism, it would at least let you know that it is a non-culturable bacteria. That is what is the strength. So, these are major applications wherein this plays a role to identify the plant pathogens. We also have, so in this case in NGS, we do not have to know the pathogen, or we do not have to know the gene which we are working on. If we do not know, we can go for a de novo, right. So, de novo means I do not have a reference at all and I do a sequencing of it. For example, fusarium, now many of them are sequenced, right, but still if I have a new strain coming up and I know it is different. As we know, the pathogens, each and every genome is very diverse and very different from each other. So, to go for a new strain, I can do a NGS. I cannot do a Sanger sequencing or anything. Sanger sequencing is when I am doing a small stretch, and for that also, if I have a genome that is beneficial, then only I can compare, align and identify the SNPs and all. But in this NGS, if I do not have a reference, if I do not know a genome also, I can do this. So, for example, there are new pathogens which are infecting your plant. So, I can do a sequencing from here and I can identify at least a closely related species, or I can identify that fine, this is a non-culturable one which is getting which is having an infection, right. So, this is what is the strength of NGS and the genome sequencing. Now, come down to the NGS processes. The basic outline of NGS processes is that we isolate DNA. When we are going for a genome sequencing, we always start with isolating the genomic DNA. I think so you had a session with isolating genomic DNA. The same process is followed. After that, we do a QC for the DNA. That also I think so you have covered. Then we have library preparation. Library preparation is nothing but ligating adapters. These are components of the library kit. For example, if you are doing a sequencing on Illumina, we have Nextera kit, we have many other kits which are available for preparing a library, right. So, these kits have been used in which we have components like adapters, which these adapters are very important components which binds to your DNA template. How will your machine identify what is your DNA template, what has to be sequenced. So, these Illumina adapters which are there, we will go in detail how they bind and all. So, those adapters attach to these DNA templates and the machine can identify that this DNA templates needs to be sequenced with the help of those adapters. Those adapters are nothing but a primer set, similar to a primer set which has a copy in the machine as well. So, it binds and then the DNA template gets sequenced, right. So, that is how the DNA template is getting sequenced. So, that is what is called as library preparation. So, when the library is prepared, the DNA templates are attached, we do a QC. Why we do a QC? We want to see what is the length of my DNA template, right. Is my library proper size or it is out of size? If I have, for example, I am sequencing 2 into 150 and if I have a library size of 1000, I will have a huge gap between two ends, right. We have a paired end sequencing. Paired end means I am sequencing from forward and I am sequencing also from reverse, the DNA template. So, when I am doing that, if I have 1000, I only have 150 and 150, it is a huge gap in between, and this gap is very important, we will see how it is important for assembly, right. So, for that we do a library QC and we know that the library of a paired end library should be somewhere around 300 to 400 base pair. This should be my library size, having an adapter of 120 bases. So, we minus it. So, we should have a DNA template somewhere around 250 to 200, yes, or else 250 to 350, wherein I have a very small insert size in it, right. Then we do a sequencing, and how the sequencing looks, we will see. We get two pairs of reads, that is R1 and R2, right, that is forward and reverse in case of paired end. So, we will see how the sequences also look. Yeah. 2 into 150 is, we have two sets of reads of 150 bases, right. We will talk about it in detail. So, how it works and what is 150 and 2. It is a read length, right. So, reads are the sequences, the DNA template is sequenced 150 bases 2 times from the forward and from the reverse. In Sanger also we have a forward read and we have a reverse read. Similarly, in this also we have a forward read and a reverse read in case of paired end. So, there are different libraries with different direction of reads, and these different directions make it very important for us for assembly, right, the gaps, filling and all. We will talk about it. So, I think so that will be getting clear. So, the library profile looks like this. wherein this is where the library is. So, in this case I have a library of approximately 814. This is a mate pair library in which it is a bigger library with a longer insert in it, right, long insert. So, if I have longer gaps in my assembly, if I have repetitive region in my assembly, this type of reads can resolve it because it has a gap between it. So, what happens in the assembly? It will consider this 814 minus 120 because 120 is my adapter straightforward. So, that goes off. 814 minus 120, that becomes my sequence insert length in between the two sequences. So, when our assembler works, it will have a overlapping or this first R1 pair and will find that much distance in between and find the other pair there, right. That is how the accuracy works. So, both the reads will move in that particular distance. So, plus and minus standard deviation will be provided to the assembler. So, accordingly it will move. So, it is not a random matching of reads, it is a accurately matching of reads like this, moves like this, right. We will see how it is. And now, in paired in, because it has a smaller distance between two reads, somewhere around 400, 495 minus 120, that is what my library is. So, you can see the library size is small. So, this is how we check the library QC on a tape station or on an Agilent bioanalyzer. This is the library which I was talking about. Aggregate each sample wise sequences. Hope you are following me. So, these small 8 bases sequences for that particular sample will be exactly identical. Based on these 8 bases, software segregate the data sample wise. Yeah. So, we know after sequencing when we get a whole FASTQ file. From there we segregate sample wise based on these small 8 bases indices. These are called Illumina indices. So, the Illumina indices is there, after that we have the sequencing adapter and then I have the DNA template. Now, Illumina sequencing chemistry is a bridge amplification. What is the bridge amplification? In my flow cell, I will have a one complementary pair of the adapter. Now, this will attach and it forms a bridge. This is how. So, this is getting attached. This. Now, for mate pair, we have a circular region. How we make it a bigger distance between? So, we have a longer distance in mate pair to resolve the repetitive regions and to resolve the ambiguity in the genome. So, if I have long gaps in the genome, if I have long repetitive regions in the genome, we use these type of libraries in which it is a circular library, right. This is also sequenced and it also has a different thing, it is a circular library and it has a junction sequence. This junction